Hello everyone, today we are going to see about the bacterial growth and bacterial growth curve. So as you can see in this particular diagram, bacterial cells divide by simple division. One cells divide to form two cells divide to form four cells divide to form 8 cells, and as it continues 16 cells, in next generation 32 cells, and in next generation 64 cells. In this way, in each cycle of replication, the bacterial cell numbers doubles, and this is called as exponential cell division. The time required for a single generation is denoted by small t. The small n indicates the number of generations. N0 is the number of cells with which you have started the experiment, and Nt is the number of cells obtained after time t. So you can see when it is one cell it is 2 raised to 0, when it is two cells it is 2 raised to 1. In next generation you will get 2 raised to 2 cells. In next generations you will get 2 raised to 3 cells. In next 2 raised to 4, 2 raised to 5, 2 raised to 6. In this way the number of cells obtained it is 2 raised to n if you are starting with a single cell. If you start with the n0 number of cells, then the number of cells obtained after time t, that is nt, is equal to n0 into 2 raised to n, where small n is the number of generations. So as you understand from this particular diagram, the bacterial cells divide by exponential way. So when you plot this number of cells with time t, you get a specific pattern of a graph which is similar for majority of the bacteria. But since the division is exponential and the values that you obtain, it goes on increasing in an exponential or logarithmic manner, it becomes difficult to plot only number of cells against time t. The time is taken on the x-axis and number of cells it is taken on the y-axis. So it becomes difficult to plot number of cells, and that's why you need to take log of number of cells on the y-axis so as to accommodate all those values on the graph. So here we will see in the next diagram how the pattern is obtained. So here you have log of number of cells on the y-axis and time is taken on the x-axis. This type of graph is obtained when you carry out the experiment in a closed system. Closed system is a system in which you provide limited number of nutrients to the bacterial cells. So as it is shown in a flask, here the liquid medium is taken and bacterial cells were inoculated. So nutrients present in the flask, they are limited. So if this is a system, you get a growth of bacteria as shown in the next graph. So this type of graph is called as bacterial growth curve, and majority of the bacteria, they show this type of growth curve. So the first phase is called as lag phase. The second phase is log phase, or it is also called as exponential phase. The third phase is stationary phase, and the last phase is death phase. So we will see the details of each phase. The first phase, lag phase, is a phase where the bacteria adapts with the environment. So you may have taken a natural sample from which you have inoculated a bacteria in a flask, or you may have transferred bacteria from one medium to other medium. So in this phase, the bacterial cells try to adapt with the new environments and the nutrients present in the new environment. The bacteria has to produce enzymes by which it can degrade the nutrients present in the new environment. So for that purpose, it has to adapt with that environment. It has to start synthesizing those enzymes and molecules which will be required to utilize the nutrients available in the new environment. So if you transfer bacteria from one medium to the same medium, you get a shorter lag phase, and if you transfer bacteria from one medium to a medium which is different and having different types of nutrients, then the lag phase that you get may be the longer phase. It depends upon the complexity of nutrients present in that medium and the genetic makeup of that organism which is inoculated in the flask. So as you have learned, lag phase is a phase where there is no increase in the number of cells. In this phase, the size of the cell may increase and cells adapt with the environment. It understands what type of nutrients are available, and accordingly it starts producing the enzymes required for degradation of those nutrients available. The next phase is called as logarithmic phase, or also it is called as exponential phase. Bacteria multiplies rapidly with a constant rate in this phase, and the multiplication rate or growth rate of bacteria is maximum in this particular phase. So as number of cells goes on increasing, you can see the increase in the log value, which you can observe in the given diagram. Then you get a stationary phase. So in stationary phase, the number of cells remains constant, or you can say total number of cells remain constant. This happens because the rate of formation of new cells is equal to rate of cells dying in that particular flask. So when you study growth curve, it is always a population. You are studying a population of bacteria and not the single bacterial growth. So in stationary phase, as I have mentioned, you will find that growth rate and cells death rate, they get matched with each other, and that's why there is no net increase in the number of viable cells. At the end of stationary phase, you will find that in that particular flask, since it is a closed system, nutrients gets depleted. So that may be the reason because of which the growth rate decreases. Second reason is accumulation of toxic components. So when bacterial cell metabolizes different nutrients, it may lead to decrease in the pH or increase in the pH, and that pH may not be suitable for the growth of the organism. So accumulation of toxic components, change in the pH are the factors which affect the growth of bacteria, and in the death phase, new cells may be formed, but the rate of formation of new cells is less than rate of formation of dying of the cells, because of which the net number of cells it goes on decreasing. So as you can see from the graph, the log of number of cells, it goes on decreasing. So these are the four main phases, as I have mentioned: lag phase, log phase, stationary phase, and death phase. So this is a typical pattern observed with majority of the bacteria, and this pattern When you plot on a graph paper, the pattern is called as growth curve of bacteria. So why to study this growth curve of bacteria? What is the significance of this bacterial growth curve? So you understand the kinetics of bacterial growth, you understand what is the generation time required for this bacteria. That means in how much time the cells divide. So smaller is the generation time of the bacteria, faster is the growth of that bacteria when nutrients are provided to that organisms. So if you want to use these cultures for fermentation industries, you always require cultures which is a fast growing cultures and can give product formation in short period of time, and that's why generation time is very much important, and study of growth curve and generation time, it is significant. So you can understand or you can predict the growth behavior, at what time it will undergo stationary phase, at what time the culture may enter in a death phase, at what time the nutrients should be supplied to the cultures in a fermentation process, and what are the effects of different parameters like temperature, pH, nutrients availability on the growth. All such parameters can be studied if you have idea about the growth curve of bacteria. Majority of the antibiotics, they act on the growing cells. So in that case, the metabolism is active, cells may undergoing DNA replication, may be involved in cell wall formations, may be involved in protein synthesis. So majority of the antibiotics, they act at these particular stages, and that's why it is important to study the growth curve of bacteria and to have an idea about at what point and till what point the cells will be present in the exponential phase. Phases of growth curves gives you idea about at what point the metabolites or fermentation products are produced. If those are primary metabolites like amino acids, sugars, enzymes, they are produced in more amount in a exponential phase. And if your fermentation products are secondary metabolites like antibiotics, they are produced in stationary or late stationary phase. So if you have idea about a time at which your culture is going to enter in different phases, you can decide the time of harvest of fermentation broth to obtain maximum amount of product. Study of growth curve or growth dynamics helps in developing strategies to control bacterial infections, whether through antimicrobial treatments or using hygienic protocols. In summary, the bacterial growth curve is a fundamental concept in microbiology with broad implications for various fields from medicine to industry, aiding in understanding, predicting, and controlling the bacterial growth.